So today we'll share some time with all of you saying about the development of bioprocess for microbial enzymes with commercial applications. And so when I given this title, that time I was not very sure that this is under the theme food and biotechnology. So however, I try to incorporate some of the informations which is relevant to the food technology as well. So, coming to the enzymes, all of us know that these enzymes are the macromolecular biological catalysts and that accelerate many of the biochemical reactions. And if you look into the prerequisites of an enzyme, the enzyme normally have an active site, that's a catalytic site to which the substrate get it binded, and that works substrate and active in a lock and key mechanism, because that is why the specificity of the enzyme comes. And the whole enzyme we call it as a hollow enzyme. It contains a protein portion, that is the apoenzyme, and a non-protein portion, that is the cofactor, which is required basically for eliciting the catalytic activity. And in some cases there will be coenzymes like vitamins. So this is how the enzymes works. As you can see down in some of the photographs, the substrate entering to the active site of the enzyme, and then how the changes, conformational changes happens once the substrate get into that one. And then there is an enzyme product, that enzyme substrate complex is forming, and then which is converted into the product and give. So this is the basic system, how it catalytically get into the substrate and convert it into the product and high specific way. So if you look into the to what you see in this slide, actually it is a house, and you can see the entire things which is there in the house. You can see the furnitures and your dining tables, computer rooms, automobiles, your pet animals. So this kind of and then the water systems. So if you look into any of our day-to-day life activities, starting from food and beverages, detergents, biopharmaceutical, wastewater treatment, or papers, pulp, paper pulp in biofuel which is required for the automobiles, and the animal health, nutrition, nutraceuticals, leather, textiles. So this all where you see how the enzymes are involved into there, and then you can see a kind of a market for all different types of enzymes in our day-to-day life. So it has been become very prominent, especially in many countries. The more enzyme-based detergents or enzyme-based activity like use of biofuels or bioenergy-based, where the enzymes has to play a key role on all the textiles, nutraceuticals, food, beverages, everywhere. Now we go into the influence of enzyme, and the demand for enzymes is keep on increasing. So this is the industrial enzyme market, which is expected to reach around 10 billion by 2024. And you can see most of the areas: food, beverage, animal feed, paper pulp, and then detergents, textiles, nutraceutical, what we discussed just now. And this 10% compound annual increment is especially for the Asia Pacific region, because there is a robust expansion of food processing industries, particularly in Asia Pacific. If you come to the Japan, Korea, Thailand, Malaysia, India, the food industries, they are flourishing like anything, and this type of enzymes is required in a very high amount. So, if you look into the major subsectors of enzyme industries, they are, I have listed here 4 or 5 major sectors: that food and feed, detergents, biofuels, which is a very upcoming area, leather and paper, and biopharmaceuticals. And if you see on the right side, the percentage revenue, how much from these different types of enzyme, what is the revenue range you getting. The latest data, global 2021, you see the food beverages from where this industrial enzymes, the market revenue coming from this area is the highest, followed by animal feed, detergents, and then you can see that biofuel or healthcare, especially healthcare and other textiles and other areas. Yes. So it is very obvious that food and feed area or the food beverages area, the enzymes applications are very high, and the revenue which is generated is also very high. So this slide I have listed some of the global very leading enzyme companies over the globe. Most of you will be knowing that the Novozymes, which is having a monopoly in many of the enzymes, especially industrial enzymes. So that is why the countries like India's many of the technologies which is based on enzymes, we are not able to make a cost effective process, because most of the times we need to use the enzymes from these kind of people. No, this Novozyme enzymes which is very much there in all they have all these enzymes. And then Genencor, DSM, Roche, BASF. So these are few enzymes or multinational companies which have the monopoly on this most of these enzymes. So countries like India have to depend upon this type of enzymes and then to use any kind of a technology process. So there is an immense scope for our researchers, our science, to look for very indigenous enzymes which can compete with these multinational things. Then only we will be able to get into the to play a major role in such kind of a business sector. So just to get an idea that most of these enzymes, and most of them are started in the early 90s, 1921, 1952, 1996, like that. In the 90s you see most of these enzymes has been flourished. And what is important, you see after 2000 there is not much big improvement. Where is the companies coming up? There is no industrial companies coming up with the this type of enzyme content, because from 19 onwards these companies started to control everything in the enzyme market. So there's a need for looking for different types of enzymes and indigenous systems, our own organisms. Then only we will be able to get into this market. This is a kind of a drawback of this particular area. So come to some of the basics. Why to look for microbial sources? Enzymes, they are there in all the living enzymes, and animals, you know if you can plants, animals, but by microorganisms. So here this slide shows some of the points that they are easy and fast growth. You can cultivate them in a bulk level. You may be knowing about the fermentation and all, where you can cultivate them under controlled conditions. They have high growth rates and contain larger proportion of enzyme in relation to their body mass than the plants and animals. Even if you want to take enzymes from animals and all, you may have to make the a particular tissue or a particular portion. Only from there you will get some kind of a enzyme, and similarly plants. But in microbes, the amount of enzymes which you can have it from there, compared to their body mass, is a very huge. And their growth is not limited by any season or climate, because we can cultivate them under controlled conditions, and we can make many kind of, and they occur naturally in a wide range of habits, the habitats, very areas. You can grow them at different at extreme temperature conditions, extreme pH conditions. I will come to those points later. And they can be easily genetically manipulated. That means you can clone a gene, a particular enzyme, and you can overexpress them, and then to make in a very huge level production of this type of enzymes. So these are all some of the advantages why we look for the microbial enzymes. And you see that it is written over here, bacteria, actinomycetes, fungi, algae, all these are known for producing various types of enzyme, like bacillus, pseudomonas, streptomyces. And fungal growth, you can have aspergillus, penicillium, trichoderma, and microalgae, chlorella. From all these microorganisms you can have the enzymes. Okay, so coming to the applications, I have already given you a glimpse showing that house, which are the things in the house. And then most of them, if you take any content, if you computer, and then you take the paper there, or you come to the dining table, you have the milk products and any kind of thing, and your automobiles, your pet animals, their feed. So that is what here also I have shown. have a role to play in industry, in environment, and in agriculture. In agriculture, you see that most of the agro processing reaction, for extraction of oils or extraction of colors, pigments, and everything from the agro residual biomass, we need enzymes. For animal feed additives, you might be knowing about the enzyme phytase, which is required very much to to cleave the phytic acid content to release the absorbable phosphorus content, and then the agrochemicals. And in the industry side, as we discussed, starch, fuel, detergents, protein hydrolysis, leather tanning, pharmaceutical, chemical, and so on. So what I said, plant or other sources, they may not have this much wide applications. You see enzymes from animals and basically for some biochemical reactions, so some therapeutic enzymes, or for community. You may not make a big alpha amylase production from a animal kind of things, or unless you clone and overexpress, that kind of things are difficult. But in the microorganisms, a wide variety of enzymes for large applications, which is possible if you look into the microbial enzymes. So I just before I told you about the extremophiles. Why extremophiles? They are organisms they have the ability to thrive in extreme environments. So they are called extremozymes. These enzymes are called extremozymes, and they are a valuable source for industry important enzymes, because that's way we can get enzymes with very particular properties, very specific properties which you cannot think of. Most of these industrial enzymes may require high temperature, some of the operation conditions, so you need a thermophilic enzyme. Some of them will be high pH, high pH in the sense, I mean low pH or high pH, whatever, highly alkaline or highly acidic. So how to do this kind of, because the point what we say for enzyme is this enzymes compared to the chemical reaction, they work in the mild reaction conditions. But even in that mild conditions, most of the mesophilic enzymes will thrive. But there will be chemical reactions which we need to do at higher temperature, or and for that also we have a solution from microbial sources. If you look into the extremophiles, so here extremophiles I have listed. What are extremophiles? Because they are either on the extreme kind of thermophile, psychrophile, that means low temperature, acidophiles, low pH, alkalophile, high pH, and then you have a piezophile, high pressure, metallophile, high metal concentration, radiophile, which can tolerate radiation to certain effect. So like that, these are the extremophiles categories, and you can see the what type of habitat it is. As per the name indicate, you have the habitat there, and there are, you can see most of these organisms, amylase, silinase, proteases, these are all able to get from extremophiles. And the same things to the geothermal site, which are the areas for this kind of extremophiles. They are the hot springs, deep sea hydrothermal, marine sulfur, or decomposed plant samples, hot springs, compost, deep sea hydrothermal vents, sediments of the hot springs, garbage dump. So if you look into this kind of very uh, hazardous or very extreme condition environmental niches, you can able to get the enzymes which can provide different types of tolerance of kind of things. You can see some of the enzymes as well as the organisms were reported shown there in this table. So you see again the biocatalytic activity relevant in extremozymes. You see some of the enzymes which is listed over here, hyperthermophilic esterases or thermophilic esterases. You see their temperature optimum is 100 degree, and half time 50 minutes it can survive at 126 degree. And similarly at 40 degree, 45 degrees, some enzymes which can survive at 64 degree for 1 hour, most of them active at 70 degree, and like that, optimum activity. If you look down, thermophilic and acidophilic amylases, optimum activity at 75 degree and pH 3. So at pH 3, getting an organism which is having lower activity, that is a kind of a lot of advantage for this. And then 4 molar sodium chloride, they are now allophilic type of kind of a culture organism. So we need enzymes like these to compete into the market or into the kind of things. So there is a need of look into the extremophiles. That is the only point which I wanted to highlight in the last 2 3 slides. So coming into the production, microbial production of this kind of enzymes, those who are in the industrial biotechnology or those who have studied this industrial biotechnology know about the fermentation. So we have two types of enzymes: submerged fermentation or liquid fermentation, and solid state fermentation. So which is a free absence of water that is there. So here it showed the typical conventional fermentation process for enzyme production. You start with a lyophilic vial, which is contains over a slant of your organism from the agar, solid agar medium, and go to into the inoculation tank, where you can optimum, to allow the culture to grow, to make to become viable, because they are coming from lyophilic conditions or from a solid agar medium 4 degree, and they have to come there and then to make the first seed of this for this production medium. And and you can see that this inoculum will go into a bigger tank, which is the main production fermenter, where you can have all these parameters to be adjusted over there, like dissolved oxygen level, biomass, and then you can supply acid, alkali, and then if it is a foam formation, you can add antifoam. So it is a kind of a bioreactor. So grow them in a bioreactor at a large level. So um, you can get a high biomass, and from you can see the desired product to get it. So this kind of thing. So this is a typical fermenter that already most of you will be knowing. So the scale of fermentation can be anywhere between a few hundred liters to as much as 50,000 liters fermenters are available. So this is one advantage of going for microbial system. You can grow the cultures in such a huge mass and to get the kind of a product, the enzyme, whatever is required. If you look for plant sources or animal sources, this kind of facilities are not there, and that is the why we look for the fermentation route, or the, I mean, microbial based enzyme kind of thing. So now come to the economics. Now, any bioprocess, it is important how effectively, how cost effective is your production. The success of any bioprocess depends upon many factors, of which the one most important factor is the raw material. What type of raw material you are using to the bioprocess, which is getting converted into the desired product. And as you showed the right side, the cost of 51%, 50 to 51% of the entire cost depends upon the raw material. And then other factors like location of the production facility, energy input, microorganism, product concentration and purity, stability, end user preferences. It's very important. See, for what purpose you are using? Are you using for a therapeutic purposes, or are you using for a technical purpose? So that depends upon the purity. And downstream is again another important cost determining factor in any bioprocess. So at what level purity you require these enzymes. So it depends upon the end use. You have the bio cost of the bioprocess will also get it changed. And you can see that microorganism is very important. Unless otherwise you have a very good potent organism, production organism, the entire process will be a failure. And also you need a very potent organism. And then location of the production facility, how effectively you are arranged your pilot plants and other kind of things. If your inoculation somewhere else and get it transported some other places for production large scale, then their transportation and many other manpower charges, so many things will come. And you see the labor 6.6%, equipment depends in general around 60 to 17%, and then the lab expenditure 2%, consumables, waste disposal. So these are all the parameters you have to consider when you go for any a kind of a bioprocess, or the scale up of the scale for a commercial kind of things. So coming to the cheap feedstock for crucial economic facility, I will come to stress a little point on the agricultural residues. I hope I am audible in between. Just want to check.
Yes, sir.
Okay. So the importance of agricultural residues, I wanted to stress, especially a country like India. We have a surplus of agricultural residues which are generated in the field at the time of harvest, or there are which generated when they are co-produced during the processing. So these residues we can classify into two: primary residues and secondary residues. The primary means in the field itself, generated in the field itself at the time of harvest. That is what paddy straw, sugar cane top, maize stalks, coconut empty, everything is the primary. But once you take it into the process, and then whatever you process, you take it, then you get the bran and paddy husk, bagasse, maize cob, coconut shell. These are all after a kind of a processing. So these are all called the secondary residues. So this type of materials are very huge, very immense in our country, which is otherwise going as a waste. We have seen about the firing of all these paddy fields and all after the harvest. Lot of environmental issues are coming up also. Why don't you use these kind of substrates? So here you can see some of the common agro residual substrates shown here: wheat straw, rice bran, wheat bran, and bagasse, especially this from sugar, and cassava is plenty is available. So after taking the starch and removal, whatever come out there in cassava, that tippi, which is called tippi, which is, it is again contains some starch, and but you can use them for many kind of productions. And all the oil cakes, you can hear the coconut oil cake, cotton seed oil cake. So these are all otherwise going little bit percentage is going for feed purposes and all. Otherwise it is a huge amount which is going unutilized properly. So there, I mean, and it has been since long time it is there, utilizing this kind of agro residues for value addition. And again, fruit waste, vegetable waste, apple pomace, grape seed waste, citrus waste, and then many seeds waste spent brewing after the beer or barley kind of thing, whatever come out, that is going out there here. And jackfruit seed, palm kernel care. So these are all substrates which is available plenty, which can be a good source for making a value addition, especially for enzymes. And also, I can tell it is more suitable for filamentous fungi, because fungi can grow on a low moisture level, and they can utilize, because they can grow the hyphae or the substrates and utilize them and convert them into the production. So many secondary metabolites, enzymes, for that we depends upon fungal. Solid state fermentation is a very good option. And see, just simply this slide showed solid state fermentation. What is happening over? You can see the growth of Aspergillus niger or nidulans or whatever, that spore formulating, and it will be completely destroyed. It will grow on the solid substrates and utilize then things and take these nutrients and make them it. So solid state fermentation or solid substrate fermentation is the fermentation process occurring in the absence or near absence of free water. And it has another several advantages which I don't want to go in a big way, because you can compare the submerged and solid state fermentation. Compared to submerged fermentation, it is more sophisticated and it need a lot of skilled manpower. But solid state fermentation, even you can have only minor technical skills are required, and not much machineries is required. And then not much water waste coming in solid state fermentation. So like that you can so many advantages you can tell about this one. But that is not under the purview of this particular talk, because this is only, I just wanted to mention the um, cost effectiveness of using agro residues. And you can see a beautiful same picture. The fungus is growing on wheat bran and spreading over there, and then this has been taken in our lab. These pictures, you can see that the wheat bran where the white fungi growing over there, and then some of the tray fermentation for the solid state fermentation where the aspergillus, you can see beautiful pictures and all. And like submerged fermentation, SSF also can be done in scale up. Different reactors are available, column type reactors, which is also known as rainbow reactor, then drums, and then koji tray reactor, like that. There is an excellent reference by Durand on Biochemical Engineering Journal on bioreactor designs for solid state fermentation. Those who are pursuing masters and other courses, they can refer. This is very good. You can get a whole idea about the different reactors. And like solid state fermentation, this also, you can have the scale up from small flask level into the tray level, and also automated systems. Or this is the rainbow model, whether you can initial moisture and other parameters for solid state fermentation can be controlled, and then again to the highly mechanized solid drums and other systems, Fujikawara Koji type of systems, which is also automated systems, is also available for this solid state fermentation now, like high volume submerged fermentation, what we are using. So this is a kind of a scale up which we did in our laboratory, in our lab, cellulase from Penicillium jandinellum. So we have cultivated 1.1, 1 kg of the wheat bran per tray, and some of the parameters which is you can control here, incubation, and then you can get a very good cellulase yield of 40 to 60 filter paper units. So you can see the beautiful trays arranged, these things, and then from flask to tray level production of this one. Okay, these are all about the production. Now come to the downstream of this enzyme production from microbial sources. Again, as I told you, this is again very crucial. Is subjected to, depends upon the intended application, and exactly I written it. You can classify this enzyme into different groups. One is technical grade enzyme. See, you wanted an enzyme for textile purposes or paper and pulp. You don't need a very high purity enzyme. What you need, a crude concentrated protein. It may able to perform the function what you are looking. So most of these enzymes, like in the pulp industry, textiles, or if you look into the some of the feed enzymes, they're all you need partially purified enzymes. So they are called mostly technical grade enzymes is enough. But if you're looking for glutaminase, asparginase, they're all therapeutic amino peptidases. You need high purity of an enzyme. So there is no other option that you have to incorporate 3 or 4 chromatographic steps to get into the purify this enzyme. Or you have to have a recombinant enzyme where you have a specific tags like his tag or something, so that you can bind to the column and easily take it out. So that kind of a purity is required. So that also depends the cost of the process, what type of object. So but industrial enzymes mostly countries, we what we focus is on the technical grade enzyme. And then the enzymes, you see, this can be either extracellular or intracellular. Extra means it is just outside in the production medium, it is available. So you have to remove the cells by centrifugation or filtration. After that, the concentration of the cell free extract, you can concentrate it is by ultrafiltration or precipitation. And if it is intracellular, you need to have one more step. You have to first you have to recover the cells and disrupt the cells by homogenization, and then you have to remove the debris, cell debris, by centrifugation or filtration. And that concentrated cell-free extract, that is called lysate basically, because it break the cells and taken, and that is also will be there. And then you can incorporate some of the stabilizers, preservatives, and adjust the biopotency, and then get to the final product. Either you can have a liquid form, or you can go to the spray dry it or drum dry, and then to get a solid powder type of things and all. Some of the feed enzymes like phytase and all, you grow them in wheat bran and get collected. And most of the time what we do, probably this fermented matter itself, you can dry it and use directly for any kind of for kind of a purpose. You don't need any kind of a purification itself. So that is the because a very crude enzyme kind of thing. So this is the steps, basic, very general steps which is involved in the enzyme, how you can take it out after the production. Okay, come to the theme. So by that time only I came to know that I have to talk it in a food section. So what I did is I did a few slides were added. Maybe it looks a little bore for you because I will be showing many enzymes. It is for what is the application, what is the kind of, but of course we, it is better I try to cover almost all areas of this food sector where the enzymes have a kind of a role. So I will quickly go for 3 or 4 slides like that, and then come to the my conclusion kind of this thing on this enzyme production. So first let us see lipase. And you know that it is an important catalyst and known for their versatile biotechnological applications, and they can catalyze hydrolysis, especially the esterification reactions, and especially they are being used for baking and oil and fuel, dairy, cheese processing, and again you can look into the mainly most removal of fat, and there are a lot of commercial products of lipases available by different names, lipopan, different lipozyme, lipase, G lipase, AY lipase, different products depends upon the companies. And you can see, as I told you, most of these enzymes are for novozymes, novonodasic amino enzymes, and all the multinational enzymes producing it. So the this is the lipase is basically used in baking industries, oil, fat industries, and cheese processing, etc. Then the phytase. So it is type of phosphatase enzyme that catalyzes the hydrolysis of phytic acid. Phytic acid is an anti-nutrient. It is not good for many of the monogastric animals, piggeries, poultry. They are not able to digest a phytic acid. So we need to release the usable form of the inorganic phosphorus, and for that need to use the enzyme called phytase. And it is also having applications in many blood processing, isolation of plant proteins, grains, especially for wet milling, and some of the strategies which is used for the probiotics and kind of prebiotic concept, cereal brand fractionation kind of thing. So there or there, the phytases has been used mainly to release the usable form of inorganic phosphorus from anti-nutrient factors like phytic acid. Okay. So come to the cellulases. Cellulases have a lot of applications. No, cellulase has been widely used for biofuel production. So what it does do? This cellulase basically play an important role in the global carbon cycle by degrading insoluble insoluble cellulase into cellulose into soluble sugars. I think that the slide that the cellulose has not come properly okay to the soluble to the sugars. So that is why. So when you use a biofuel like bioethanol, the idea is to generate fermentable sugars from complex like no cellulose or hemicellulose. So cellulases is an enzyme complex which is playing there, and then it has a role in de-inking or anything related to paper pulp and other things. But what we discuss here is its role in food industry. So cellulase is used in combination with the pectinase, hemicellulase, and combination of these three enzymes, especially cellulase, pectinase, and hemicellulase. They are known as the macerating enzymes. So macerating enzymes is basically is used for fruit processing, fruit juice processing, and fruits in their natural form tend to get it rot. So we need to make the pulp and store it for improving the shelf life. Okay. So the addition of this macerating enzymes during the process can reduce the browning. That is a kind of conformational changes which happens to the juice, which is not very good, and it is used to improve degrading structural polysaccharide that interface with the juice extract clarification and its filtration and so on. And also it is used in the extraction of olive oils, and this resulted in increased extraction, better quality, more antioxidants and etc. Olive oil is an important ingredient in the food, right? And the alpha amylase already everybody knows it is a starch degrading enzyme. Alpha 1 4 glycosidic bond has been broken by this amylase, and it has a vast role to play in the processed food industry: baking, brewing, preparation of digestive, production of cakes, fruit juices, starch syrups, and other different alpha amylase and glucoamylases. So these are all starch degrading enzymes, and starch degradation is important steps in the food industry. And comes to the amino peptidase. So this is a very interesting enzyme. This is again belongs to the exoprotease family, which can catalyze the cleavage of a peptide bond which contains a particular amino acid. So it can cleave and remove one particular amino acid from the N terminal. If it is removing leucine, it is known as leucine aminopeptidase. If it can remove proline, it is known as proline aminopeptidase. So substrate specific enzyme, kind of an very interesting exoproteases enzyme. And this enzyme can widely used in food industry, especially for removing some of these amino acids which cause bittering into the food product. So for the debittering of in cases in baking, brewing, cheese baking, they are used for removing the bitterness of the peptides, because they can remove some of the amino acids, especially leucine, which is very widely used in the protein hydrolysis preparation. When we make it that kind of things, to remove the bitterness. Okay. Okay, now come to the lactase, which is required for hydrolysis of lactose, which is and again an important enzyme, biotechnological enzyme for in food industry. It is for release the formation of galacto oligosaccharides, and these galacto-oligosaccharides could used as a prebiotic for various food ingredients. And you can see some of the applications of application of this enzyme: enhancing shelf life of lactose containing material, lactose hydrolysis in milk, GOS production, and kind of applications over there. And pectinase, again, very wide applications. You can see fruit juice extraction, brewing industry, oil extraction, paper and pulp application, coffee, tea, paper making. So, and also the pectinase is basically it has been used for hydrolysis of the glycosidic bonds in pectin polymers, and this is improved removal of to make the clarify the fruit juices when it is being prepared. It has been widely used in shape. And phospholipases used in the processing of various dairy products to enhance the stability of the fat and to maximize the yield of cheese, milk, butter, and ice cream. Similarly, glucose oxidase, this is also known as oxidoreductases and has many role in food industries. You can see that some of the role, color enhancement, flavor enhancement, increasing storage life, aroma, stability of the food products, like that it is there, oxidoreductase. And comes to the other categories, which is silanase, which is very much application in paper and pulp industry. Silanases, they have been used for that. But here we require to improve the texture, tastiness, and palatability of biscuits. Role of juice production by improving the extraction, clarification, and stabilization. And then in beer making industries, it has been widely used with along with arabinosilans and lower the oligosaccharides content to improve the viscosity of the beer. Okay. And then the catalysis, especially for food preservation and to eliminate the peroxidase from milk, to remove glucose from egg white, baking industry, and in food wrappers to prevent oxidation control, and also used for the packing as sub packing materials and other things. And I quickly go for 2 3 other enzymes also: peroxidases for producing flavors, color, texture, and improving nutritional quality. And invertases, it is a confectionery, it is a catalytic agent in obtaining artificial sweetener in inulinase, invertases, and you can make artificial sweetness. It's having huge market now because all people are diabetic. We need artificial every day. Everybody need 3 types. So this has been very much required for this making the artificial sweetener. And then asparaginase is a very interesting enzyme. What is the this asparaginase, but this one asparagine is converted into acrylamide when we buy all these chips and other fried items from the bakery and market and all. And it causes, when you buy this frying and other thing, this asparagine is converted into acrylamide, which is a carcinogen. And that is what we eat, very crispy and kind of things, and they will do it in 10 times, and then we buy this one. But anyway, we this asparaginase can reduce, it can reduce 97% of the asparagine in this kind of thing, and then this acrylamide formation can be reduced. And naringinase is again a debittering enzyme, especially for the citrus fruits. You have lot of jams and other, many you orange and other lemon and kind of things, many jams has been used, and there this naringinase is used, the debittering enzyme, and also it is a sweetener and tomato pulp preparation. So okay, like that I can go list maybe another 10 enzymes which is used in the food industry, but that will be very boring and kind of a bad treatment for all of us if it is there. So I summarized everything over this particular table. You will see the food industry, the major applications: dairy, brewing, alcohol production, baking, wine and fruit juice, meat, protein, starch, inulin, beverages, cassava, fermented foods, and everything. And you see most of these enzymes which I have discussed with you: protease, amylases, cellulases, glucanase, amylase, everything has been used in the thing. And the purpose also very briefly I have covered all these. So we will leave from this topic, and a couple of slides for my institute, my division. We have working on microbial process and technology, and which is relevant to here is our activities related to the industrial enzymes. We have been transferred the technology to various industries. Some of our enzymes has been literated is here: alpha amylase from Aspergillus oryzae, and phytase from Ficum, Aspergillus ficum, and cellulase from Penicillium gentianellum, and a silenase from Bacillus pumilus, and all of them have a very good activity. And this has been used to different companies on a non-exclusive, like first enzyme transferred to Capis Biotech Mysore, and MAPS enzyme, which phytase has been transferred, and they have also said this is one of the good enzyme that they got from us. Still, they are using our culture for the MAPS enzyme, which is one of the major enzyme playing company in India, which is located in Ahmedabad. And this Thermax, they are using the cellulase kind of things. So we could make this kind of enzyme because of this kind of facility which we have created in our division. As I told you, we have a 2G ethanol pilot plant over here, because basically we can treat 80 kilogram of the biomass, and from that you get the ethanol. And for that pre-treatment, hydrolysis, all the unit operation can be done in our pilot plant. And we have the fermenters of different capacities, starting from a parallel system for 500 ml to 150-liter fermenter, large scale fermenter, which is available, 3 liter, 20 liter, 150. And come down, you can see the solid state fermentation pilot plant, which is a Koju room. And you can see the controlled humidity and temperature, every other factor, everything controlled. And you can keep the trays on that specific room, and you can perform your solid state. You can see the periclave, very big autoclave, where you can put the trays, can be put it in there, get it autoclaved and get into this kind of thing. So this kind of facility is very essential for if you want into a commercial scale. Even then, our facilities are not sufficient. Now, companies are coming and asking, they wanted a 1,000-liter demonstration or 5,000 demonstration, and we are not able to show to the company, and unless otherwise they can get convinced that large scale production. Because when you scale up, a lot of changes will happen. So, for example, solid state fermentation, you do it in a flask, you will get a high product. But when you go to the 500 gram or if you 1 kg tray level, the entire scenario will change, because you have to adjust the temperature, bed weight, how much of the substrate can be put up, because it's all depends on the mass transfer, the how much aeration, how much other kind of nutrients reaching, how homogeneous it is, because SSF can be very heterogeneous. So how good it will be there, and then somewhat fermentation also have its own problems, contaminations and large water conditions and kind of things. So facilities and maintenance is very important for any laboratory to get into this kind of work. So come, let us discuss a little bit about the recent developments in enzyme technology. So the overall goal of the innovation of the enzyme manufacturing industry is, you can classify into three points. That is exploitation of new types of enzymes, improvements of enzyme properties, and efficient production processes. So these three factors are very crucial. No, you need to have a new types of enzyme. I told you, the no, when we need some processes, you need an enzyme which are worked at 100 degree, or it may can go at 3 pH 3, and this kind of improvements in the enzymes is required. And the production process, the production, no, you need raw material which is very smart. You can use a glucose, you can use a synthetic medium, very high large, but if you see yeast extract, beef extract, all those things you add, organism will be happy. But no, you will not be able to sell this item, because people don't want to use this kind of, no. So there is a very interesting topic, defined medium and undefined medium. Defined medium is a synthetic, you know that all these ingredients are there and use it. But what we have to do out of box, we have to look, we may use sometimes fish meal extract, or some marine kind of a waste materials, where we need to supplement the enzymes like that. And then the agro residues, which I mentioned a bit. Okay. So although the considerable improvements had been made in process engineering, large scale fermentation and downstream purification of the enzyme remain to be significant in bottleneck innovation. So still there we need to improve our downstream and kind of things. And uh, the introduction of genetic engineering is the most, probably the most important breakthrough in enzyme manufacturing. Most, almost all these companies, what they are using is engineered organisms, and they will have a very good expression system to put this enzyme. So the contribution of genetic engineering into this one is reduction of manufacturing costs. This increases of enzyme yield by increasing the enzyme expression in the production organism. No need of a de novo design or production process for a new production organism. Instead, the enzyme gene of interest can be cloned into a well-known production strain, and then you can have overexpression of this enzyme. So you can increase the titer of this enzyme. And then new enzymes we need, increase the accessibility of new enzymes, especially from extremophiles, and the isolation of the respective genes and expression non-production states. That's why in metagenomics, if you look into, there are culturable and unculturable strains. Many unculturable organisms are there, or their DNA is available. You make library of that one, and you may get a very good gene which is can have a very high, you, if the gene is okay, you can get it, clone it, and then or produce it. That is the beauty of looking for microbial organisms. Options for genetic engineering is available. And the improved enzyme proteins, rational protein engineering, directed molecular evolution, these kind of things are improved. And the improved product safety, you need to look into the safety organisms which can be used. See, pathogens are there, and that cannot be used it in a industry level. There is a salient features of industrial organisms, fast growing, and genetic tools available, and they should not be highly pathogen like that. And so you have to use the safety systems and kind of things. So now the science has been developed, bioinformatics has come, protein chemistry, genetic engineering. So all these things has come together now for us for engineering the protein, or protein the enzyme. So basically the three approaches which is exist for generating enzymes with improved properties: oligonucleotide directed mutagenesis, where the enzyme properties can be modulated by exchange of a single or few amino acid residues. That is kindly done by this one. Or second one, you can the exchange of functional domains. glucoamylase and alpha amylase related enzymes. Their functional domains can be exchanged, and you can make a hybrid enzyme. So you can cleave 1,4 and 1,6. Both can be done by this kind of enzyme. This can be kind of things. Hybrid enzymes can be made by DNA shuffling systems and other things, and then you can activate the site of an enzyme. Can be introduced in, say, a small protein fragment which it doesn't have what is required enzyme. Required is an active site. This active site you can put it into a fragment protein scaffold and make it a small protein fragment can be worked as an enzyme. So like that, plenty of options are coming up now, and now this crisp technology, how we can improve the properties, enzymes, and kind of things. So this ample options are available now to look into the microbial process. So I will stop here. I think it is 4:30. So for 5 minutes more, because I had a small sorry to say that I cannot come on video today, because that's why I little bit delay in the morning. I have some issues with my video, so I am sorry for that. I would like to come on video, but because I have to change my laptop actually and come on my desktop, and there is no webcam right now. So sorry for that. So I will conclude. The industrial enzymes market will grow steadily, mainly due to the improved production efficiency, resulting in cheaper enzymes, new applications field, and new enzymes from screening organisms by engineering, and techniques such as protein engineering, gene shuffling, and directed evolution will enable development of enzymes better used for industrial elements. Then third point is tailoring enzymes for specific applications will be future trend, with the continuously improving the tools, further understanding structure function relationship, and increase the searching for enzymes from the exotic environments and kind of things. So this is my last stat. Once again I would like to thank you, the organizers, for giving me this opportunity. And this is our division, NIST, and you can see the pilot plant of biofuel plant over there. And this is my group working on a couple of them still working on this enzymes system. So thank you so much.